Hello friends, welcome to my course on Microbial Biotechnology. We are in module 6 discussing the industrial and pharmaceutical applications of microorganisms. In this lecture, we will be discussing about engineered microbial consortia for industry. This lecture is divided into 3 broad sections. In section 1, we will discuss first what is a microbial consortia, then we discuss what is the importance of such microbial consortia, and we discuss the interactions between various strains in a consortia, the special interactions in microbial consortia, the types of microbial consortia, natural versus synthetic consortia, then advantages of microbial consortia, the synergistic interactions, resource sharing, and the challenges in engineering microbial consortia, the stability, communication and control, a comparison with monoculture systems, advantages and limitations, methods to reconstruct synthetic consortia, strategy based on rational design, adaptive evolution, comparison of the adaptive evolution strategies. Then we also will discuss the methods to construct synthetic consortia, source based, top down approach, bottom up approach, and comparison of top down and bottom up approaches. We will come to the details of the other 2 sections as we pass by. First, let us see what is the idea of a microbial consortia is. A microbial consortium is a structured community of 2 or more microbial species or the same species, but with different strains, in which members will interact and cooperate to perform specific functions that individual species might not achieve as effectively on their own. These interactions can be symbiotic, mutualism, commensalism, or competitive, antagonism or parasitism. So, here we can see the categories of microbial consortia based on the characteristics of their formation. First one is the natural community, which we isolate from soil or say for example from compost, and from there we can obtain the defined consortia, whereby we do the metagenome analysis, and then we other way around do not do any characterization. So, we use the undefined consortia. So, here the production capacity of such undefined consortia will be unknown, and in the artificial community we have pure cultures. You can see different colors depicting different cultures or maybe different strains. From there we select say 2 microbes A and B or 2 strains, and then we constitute them into a consortia, and we know the production capability of these artificial consortia in advance. Then we have synthetic community, where we have a microbe A and a microbe B. The microbe B is changed by genetic engineering. Maybe we have transferred some gene over there which produces a heterologous protein, and then we mix both the natural microbial strain A and the engineered microbial strain B, and then constitute the consortia. Here the production capability is enhanced due to this genetic engineering step taken in the development of the consortia. So, to sum up, we can have natural community, artificial community, and synthetic community. Now, based on the above discussion, we can see that there is a natural and synthetic consortia. In the natural microbial consortia, these communities evolve through ecological and evolutionary process. In the synthetic microbial consortia, these are artificially designed microbial communities engineered for specific industrial or biotechnological purposes. In the first case, they are self-regulating and adaptive to environmental changes, composed of diverse microbial species found in soil, oceans, human gut, wastewater treatment plants, etc. Some examples are the rhizosphere microbiome, plant root associated microbes aiding in nutrient absorption. Then we have the human gut microbiota, essential for digestion and immunity. Then we have the wastewater treatment consortia, which decompose organic pollutants. In the case of synthetic microbial consortia, These are constructed using genetic engineering and synthetic biology. These are designed for tasks like biofuel production, bioremediation, and pharmaceuticals. They require controlled conditions for stability. Examples include engineered yeast bacteria consortia for bioethanol production, synthetic probiotic consortia for gut health improvements, bioengineered bacteria for heavy metal bioremediation. Now, what is the importance of microbial consortia? They provide enhanced metabolism. Such consortia facilitates syntropic interactions for efficient nutrient cycling, example anaerobic digestion. They provide environmental resilience, improves adaptation to stress, example biofilms in the extreme environments. They can be used for bioremediation, degrades complex pollutants, example Pseudomonas bacillus consortia for the oil spills. And these consortia have industrial applications whereby they enhance biofuel production, fermentation, and waste treatment. Then they provide medical benefits, for example, supports gut health, example probiotics, and disease prevention. They are also useful in agriculture where they boost plant growth, nitrogen fixation, for example, rhizobium and azospirillum, and biopesticide efficiency. So, once we constitute these microbial consortia, or we put two microbes of different species or different strains together, what could be the interactions between these two entities? So, number one, there may be no any interaction between them that we call as neutralism. Then there may be one-way interactions where A impacts B, okay, that is a commensalism, and it may also leads to amensalism. Then there may be two-way interactions where in the case both A and B influences each other or interacts with each other in a positive way, so that is mutualism. Or there may be competition between them, or in another case one species may benefit the other, but the other actually may predate over it, that is predation. So, in this you can see the various interactions between the strains. In one you can see there is a strain A and B in neutralism. The growth kinetics is different when they are co-cultured and when they are cultured alone. For example, in this case when NEA is co-cultured, here the strain is growing at a much lower rate than it used to grow when it is grown alone. So, this is some kind of a neutralism. As a control, NE-A and NE-B grew equally well alone and together, confirming neutral interaction apart from the nutrient competition. In the case of amensalism, in number 2, you can see AMA produces nisin, killing AMB, while both grow alone. Only AM survives in co-culture, confirming its toxic effect. Then comes the third relationship, commensalism. Here you can see CMA this grows alone in tetracycline media due to built-in resistance, while CMB cannot. However, in co-culture, CMA's nisin production activates CMB's resistance, enabling its growth. Both strains compete for nutrients, reducing CMZ's growth in co-culture. Then we have the bidirectional interactions. In the case of 4, you can see two strains Co-A and Co-B split the task of producing active nisin, which grants tetracycline resistance. Neither strain grew alone, but both thrive in co-culture, confirming cooperation was essential. Then in the 5th case, there is a competition. The two strains, competitive strains CPA and CPB produced competing antimicrobial compounds. CPA outcompeted CPB. So, altering bacteriocin production changed competition outcomes, including close contest and CPB dominant. In the case of 6, we have the predation. One strain PRA provided tetracycline resistance, while another PRB harmed it with a toxin. Initially, PRA grew, but PRB eventually dominated, confirming a predator-prey dynamics. Let us now look into the spatial interactions in microbial consortia. So, this is a biofilm with many bacterial species over here, and you can see the exopolysaccharide matrix over there. And in the case of B, you can see these are fungi to which some bacteria are adsorbed or attached. So, biofilms are microbial communities which are enclosed within a matrix and attached to surface or interfaces designed for bioprocessing and biotechnological applications. In the symbiotic relationship, the interactions occur among various microorganisms, including filamentous species like the fungi and other microbes such as bacteria, and also sometimes algae through the surface attachment. Now, in another example of special interactions, we have cell-cell interactions where there is a chemotaxis product. So, this involves the chemotaxis responses triggered by diffusible molecules which are secreted by one, and then electrostatic interactions as you can see over here due to the surface charges and the formation of nanowires or nanotubes for transporting essential nutrients and communication signals like electrons and protons. So, here the bacterial cells are trying to, of different strains or different species, are trying to communicate with one another by either some kind of electrostatic interactions or by sending in some molecules or establishing physical contact through nanowires. Then we have the endosymbiosis. Here one or more prokaryotic species reside within a host cell, forming a close biological relationship. So, there is a big eukaryotic cell which is now hosting a prokaryotic cell, and they exist in a mutually beneficial symbiotic relationship. So, this we call as endosymbiosis because one species is inside the other species. So, what are the advantages of microbial consortia? They provide synergistic interactions due to which an enhanced metabolic efficiency is achieved. Microbes complement each other's metabolic pathways, improving productivity. Then there is increased resistance to stress. Consortia are more resilient to environmental fluctuations compared to single strains. Then there is improved process stability. Cooperative interactions help maintain stability in industrial and natural environments. Then there is a optimized bioconversion opportunity. Complex substrates are degraded efficiently by different microbial species. The second advantage is that of resource sharing. There is a cross feeding. One species produces metabolites that another species utilizes. Example, lactic acid bacteria producing nutrients for yeast in fermentation. Then there is a division of labor. Different microbes specialize in specific tasks, increasing the overall efficiency. Then there is reduced competition for nutrients. Microbial species can coexist by utilizing different resources, preventing competitive exclusion. There is better utilization of substrates. Diverse microbes processes a wider range of raw materials, maximizing conversion efficiency. What are the challenges in engineering microbial consortia? The first and foremost issue is the stability issue. Mutations, population imbalance, environmental fluctuations, and unintended evolution can disrupt function. The solution for this is adaptive evolution, monitoring genetic safeguards. Then there are the communication barriers, disruptions in quorum sensing, crosstalk, and inefficient metabolite exchange hinder coordination. The solution to these communication barrier problem is engineered signaling and synthetic biology tools. Then there is the challenge of control constraints. There is a difficulties in regulation, containment risks, and scalability challenges which affect reliability. The solution for these can be found in the CRISPR based control, inducible gene expression, and physical containment. Now, let us compare the microbial consortia with monoculture systems. They have an advantage over monocultures. Here there is enhanced metabolic diversity. Consortia can perform complex biochemical transformations that single strains cannot achieve. There is increased process efficiency. Division of labor among the different microbes optimizes substrate utilization. There is improved environmental stability. Consortia are more resistant to environmental fluctuations than monocultures. There is better waste degradation and bioremediation. Multiple species work together to break down diverse pollutants. Then there is a reduced risk of contamination failure. If one species fail, others can compensate, ensuring process continuity. But in the case of single culture, we do not have this luxury. So let us have some example. For instance, in biofuel production, microbial consortia allow for efficient lignocellulose breakdown and fermentation, whereas a single strain may not handle all steps effectively. What are the limitations over monoculture? When we see that certain microbial consortia may be difficult from the point of view of complex regulation and control, managing interactions, and stability is more challenging than with a single strain. Then it is difficult to maintain population balance. One species may outcompete others, disrupting efficiency and thereby transforming it into a single strain after few rounds. Then there is a genetic and evolutionary instability. Mutations and horizontal gene transfer can alter microbial behavior over time. And it is also harder to scale them industrially. Standardizing and optimizing mixed cultures for large scale production is a very complex process. It also suffers from higher costs involving monitoring and optimization. So it requires advanced synthetic biology tools and continuous process monitoring. So some examples, for instance, monoculture systems like yeast fermentation for ethanol production are simpler to control and scale than multi-species consortia. What are the various methods in our dispensation to construct synthetic consortia? The number one is the strategy based. Based on the strategy of development, microbial consortia can be developed by two methods. The first method is rational design approach. Here, engineering microbial consortia is done using synthetic biology, then computational modeling and genetic modification. The other is the adaptive evolution approach, where we allow microbial consortia to evolve under selective pressures to naturally develop desired traits. So, in a rational design approach strategy, we employ genetic engineering, which is the direct modification of microbial genomes to introduce desired traits. We also go for metabolic engineering, whereby we design pathways for optimal resource sharing and division of labor. Then we go for quorum sensing control. Here, engineering microbial communication for coordinated behavior. Then we also use synthetic circuits by incorporating gene circuits to regulate microbial interactions dynamically. For example, engineering Escherichia coli and yeast to work together in biofuel production, where E. coli breaks down biomass and yeast ferments sugars into ethanol. So, what are the advantages of rational design approach strategies? It is precise and have control over microbial functions, predictable behavior and optimized performance. However, the challenges that are associated with rational design is the complex design strategy, high costs and potential instability in dynamic environments. Let us discuss about the adaptive evolution approach strategy, where we go for directed evolution. Here, repeated culturing under controlled conditions favor beneficial interactions. So, this was discussed already in the earlier lecture. Then we again go for serial passaging, where we gradually expose microbes to specific substrates or environments to enhance cooperation. Then we go for natural selection in bioreactors, cultivating microbial consortia in industrial setting to evolve stable, efficient communities. So, some example of adaptive evolution approach strategy includes evolving microbial consortia in wastewater treatment plants to improve degradation of emerging pollutants. So, what are the advantages of adaptive evolution approach strategy? It offers greater robustness, adaptability and suitability for dynamic environments. However, it also suffers from various challenges, like it is a very slow process, it offers less precise control and potential unpredictability in evolved strains. Let us compare some of the adaptive evolution strategies like directed evolution, serial passaging and then natural selection in the bioreactors. So, when it comes to selection mechanism, directed evolution is a human directed screening or selection for specific traits. In serial passaging, we go adaptation through periodic bottlenecks and environmental shifts. In the natural selection in bioreactors, we go for environmental conditions which drive natural selection. From the point of time scale, directed evolution is short to moderate, takes weeks to months. Serial passaging is moderate, weeks to months. And natural selection in bioreactors is actually long term process, it may take months to years. Then from the point of control view, in directed evolution, its control level is high, specific traits can be targeted. In serial passaging, it is moderate, selection pressure is applied indirectly. In the case of natural selection, the control level is very, very low, it depends on environmental factors. The source of genetic variation in the case of directed evolution is induced mutations, recombinations or rational designs. In the case of serial passaging and natural selection in bioreactors, it is natural mutations and recombinations. In natural selection in bioreactors, we can also have horizontal gene transfer as well as competition. So, what are the advantages of directed evolution? It is highly specific and targeted improvements can take place over there. In serial passaging maintains community stability while allowing adaptation. In natural selection in bioreactors mimics natural ecosystem and also long term evolution. However, there are various challenges. For example, directed evolution requires expertise and screening tools. Serial passaging, there is a risk of genetic drift or loss of diversity. In the case of natural selection in bioreactors, it is hard to control and predict the outcomes. Now, let us look into the methods to construct synthetic consortia, source based. So, these can be developed by two methods. One is the top down approach, where engineering microbial consortia using synthetic biology, computational modeling and genetic modifications take place. Then there is the bottom up approach, where we allow microbial consortia to evolve under selective pressures to naturally develop desired traits. So, let us discuss the top down approach in microbial consortia development first. The top down approach involves selecting and optimizing naturally occurring microbial communities by applying selective pressures such as changes in pH, temperature, nutrient availability and oxygen levels. So, as you can see, it is a top down approach which starts at the top. So, here naturally occurring microbial consortia are first taken, then we go for applying selective environmental parameters, then we go for the characterization of the microbial community and we try to achieve the stable consortia which we will be using for our further work. This method relies on ecological engineering to enhance the growth of beneficial microbes without requiring detailed genetic modifications. The microbial community is sourced from environments such as soil, wetlands, compost or animal guts, example rumen microbiome, are adapted to desired functions such as bioremediation and biofuel production. In the case of bottom up approach in microbial consortia development, we focus on designing microbial consortia from individual strains with known genetic and metabolic traits. So, we start from the bottom, where we take various well characterized strains and we design and assemble these into a consortia, and it involves ecological and metabolic engineering, whereby we obtain the stable microbial consortia. Synthetic biology enables precise metabolic engineering to optimize pathways for industrial applications such as biofuel production and pharmaceutical synthesis. Computational models help predict microbial interactions, ensuring stable and productive consortia. Now, let us compare these two approaches, which one is having what kind of advantage and disadvantage. From the point of view of starting point, top down approach takes natural microbial communities, bottom up approach takes isolated engineered microbial strains which are available. From the point of modification, in top down approach, selection is via environmental manipulation. In bottom up, it is rational design in synthetic biology. From the point of view of control of a community, in top down approach it is low to moderate, in the bottom up it is very high. Point of view of implementation complexity, top down approach is having less complexity and it relies on adaptation. Bottom up approach, it's having higher complexity, requires genetic and metabolic engineering. From examples we can see in top down bioremediation, wastewater treatment, in bottom up biofuel production, industrial biotechnology. Let us now go to section number 2, where we will be discussing about the synthetic biology in microbial engineering, control points for microbial consortia, strategies, then division of labor, spatial and temporal organization, community interactions and environmental influence, interactions through quorum sensing, enhanced interactions, feedback loops, environmental and nutrient conditions. Then we also discuss about gene circuit design for microbial communication, modular approach to engineering microbial consortia, metabolic engineering for efficient bioprocessing, computational modeling of microbial consortia. So, let us start with synthetic biology in microbial engineering. So, synthetic biology in microbial engineering integrates biology, engineering and computational design to create microorganisms with enhanced or novel functions. So, we use various gene editing and genetic engineering tools, for example, recombinant DNA technology, whereby we insert, delete or modify genes. Then we use genome editing tools like CRISPR-Cas9 for precise manipulation. Then we also use gene circuits, which are synthetic pathways for controlled functions. Then we use tools of metabolic engineering, for example, pathway optimization, enhanced biosynthesis of desired compounds. Then we go for synthetic metabolomes, which are engineering enzyme clusters for efficiency. Then we go for flux balance analysis, where computational modeling of metabolism is done. Then we use microbial communication and control, where concepts like quorum sensing engineering, designing microbial signaling systems, then synthetic promoters, fine tuning gene expression, then optogenetics, light control gene expression, etc. are adopted or used. So, these are overall a overview of the tools and techniques in synthetic engineering. We are providing here only few. There are many which we will be discussing from time to time. So, what are the control points for microbial consortia and the strategies? There is an input which helps in the division of labor, and then there is a spatial and temporal organization, there is a community interaction and environmental influence, and then there is a feedback loop, environmental and nutrient conditions. Then this leads to the synthetic microbial consortia. Number one important thing is the division of labor. By controlling microbial interactions, we can design more stable and productive microbial consortia for biotechnology applications. Microbial consortia are often more efficient than monocultures because different microbes specialize in different specific tasks. This reduces competition for resources within a single cell and prevents accumulation of metabolic intermediates that could slow down reactions. By assigning different functions to different microbes, we can improve efficiency, stability and productivity in engineered microbial communities. Now, here you can see in the first case undivided metabolic labor. There is asymmetric competition for intracellular resources, and there are then these intermediates accumulates to high concentration. Then there is a divided metabolic labor. There is no competition for an intracellular resource. So, intermediates maintained at low concentration. Then comes the spatial and temporal organization. Microbes can be arranged in different locations to separate incompatible processes. For example, aerobic and anaerobic microbes can exist in the same system, but in separate layers to avoid competition. Scientists use barriers to position microbes in ways that sometimes enhance beneficial interactions. So, we can see this in the Microbial Consortium. Number 1, exhibiting interactions among the populations, special configurations affect the intensity of these interactions by regulation. Then in number 2, we can see the proximity between distinct populations on a substrate, or in number 3 by interposing a physical barrier as you can see over here that segregates different populations. The thickness of the arrows denotes the intensity of these interactions. So, in temporal organizations, some microbes or processes work better in different time phases rather than being in the same time, same space. This is seen in nature, such as nitrogen fixing cyanobacteria that alternate between photosynthesis and nitrogen fixation at different times to avoid interference. So, microbes interact through physical contact, chemical signals or quarant sensing or metabolic exchanges. So, as a community, they have a lot of interactions and also influenced by environment. These interactions can change based on environmental conditions, shifting relationship from cooperation to competition depending on nutrient availability. Understanding these dynamic behaviors helps scientists to design synthetic consortia with predictable and controllable properties. What are the interactions through quorum sensing and how it takes place? Dunker in 2021 reported a synthetic predator-prey system using E. coli strains that communicate via quorum sensing. The predator strain constitutively expresses a suicide protein CCDB, but survives when the prey produces QS molecule 30C6 that induce an antidote CCDA. Conversely, the predator produces a QS molecule 3OC12 that triggers CCDB expression in the prey, controlling its population. Depending on inducer concentration, the system exhibited 3 dynamics: prey domination, oscillatory predator-prey cycles, or predator dominations. So, here in this figure you can see the example of a synthetic microbial community engineered to exhibit a two-way interaction via QS. The predator prey CC system, CCDB, is here, which is a suicide protein, and then you have this antidote CCDA. So, as already discussed, depending on the inducer concentration, this system will exhibit 3 different situations: prey domination, oscillatory predatory prey cycles, or predator domination. So, let us discuss about the enhanced interactions. So, here researchers have expanded this predator-prey model by introducing additional strains and interactions. So, the enhanced predator-prey system, the prey deactivates antibiotics, thereby protect the predator. Predator produces bacteriocins, killing the prey. With an invader strain, if the invader benefits from the prey but is killed by the predator, predator-prey oscillations persist and the invader dies out. If the invader is killed by the predator but does not benefit from the prey, both predator and prey will die, allowing the invader to thrive. These models demonstrate how quorum sensing and gene circuits regulate microbial interactions, aiding in the design of synthetic consortia for biotechnology and medicine. Then there is a role of the feedback loops, which are regulatory mechanisms in biological systems where the output of a process influences its own activity. Essential in microbial consortia for controlling population dynamics, optimizing metabolic pathways, and ensuring cooperative interactions. So, we have positive feedback which amplifies beneficial interactions. In Vibrio fishery, the autoinducer AHL activates the Lux operon at high cell density, enhancing its own production in a self-reinforcing loop, leading to synchronized bioilluminations. Then there is a negative feedback loop that regulates population balance. For example, Scott et al. Showed that using synchronized lysis circuits in E. coli allows both strains to limit their growth, enabling them to survive together by taking turns in population control. So, here we can see negative feedback. Lux R and RPA R are quorum sensing systems. Each strain lyses only when its specific autoinducer 3OC6 or PC reaches a threshold, enabling coexistence despite differing growth rates, and this figure we have adopted from the work of Dunker et al. Let us discuss about the environmental and nutrient conditions. The stability and efficiency of synthetic microbial consortia depend on precise control over environmental and nutrient conditions. Factors such as pH, temperature, oxygen levels, and substrate availability play a crucial role in optimizing metabolic interactions, preventing dominance by one strain and maintaining functional balance. Precise manipulation of these factors enables predictable interactions and maximized productivity in biotechnology applications. For example, we have in this case by Sabab, they use a membrane aerated bioreactor to establish an oxygen gradient and enable a 3-strain consortium for lignocellulosic biomass conversion. Let us now look into the gene circuit design for microbial communication. These gene circuits are engineered systems within microbes that control gene expression and cellular behavior to perform specific tasks. Genetic circuit design for microbial communication involves engineering regulatory pathways that enable microbes to exchange signals, coordinate behavior and function collectively. These circuits often mimic natural quorum sensing systems, but are fine tuned for specific applications using synthetic biology tools. What are the components of a gene circuit? Promoters come in number 1, DNA sequences that control the transcription of genes by facilitating RNA polymerase binding. Then we have the ribosome binding sites. These are short nucleotide sequences upstream of the coding region that facilitate ribosome attachment and the initiation of translation. Then there is the protein coding sequence or CDS. This encodes the desired protein and must be optimized for expression in the chosen host. Then the terminators, which are the sequences that signal the end of transcription to prevent unintended read through. So, these figure you can see the basic components of a genetic circuit, the promoter, ribosome binding site, then protein coding sequence and the terminator, which are together constitutes the insert, and this is cloned into a vector which may be a plasmid. What is the modular approach to engineering microbial consortia? Modularity refers to designing biological functions as independent and interchangeable units that can work in different settings. It allows for rapid and efficient information transfer, enhancing the control of circuits. In gene circuits, modulation can involve adjusting components. So, here you see there are 4 modules 1, 2, 3 and 4. The module reconfiguration is important to correct failures or improved performance, such as changing host conditions or replacing toxic components. It divides the microbial functions into independent reusable modules, enables scalability, flexibility and predictability in synthetic consortia. Now, let us discuss about the metabolic engineering for efficient bioprocessing. Metabolic engineering focuses on optimizing the production of metabolites and biomolecules traditionally using single strains. However, synthetic microbial consortia are increasingly being explored to divide genetic circuits and metabolic pathways among different microbial populations. This division helps reduce metabolic burden on individual cells and improves efficiency for complex pathways. So, let us now look into some example where Cha et al. Designed a mutualistic microbial consortia for stable conversion of carbon monoxide to produce value added chemicals. So, in this picture you can see carbon monoxide going through the cell and then converted to acetate, which is then transferred to another cell in the consortia, which will be converting it into different value added products. So, these Escherichia limo, some naturally consumes carbon monoxide, but accumulates acetate, which is inhibitory. So, engineered Escherichia coli converts this acetate into valuable biochemicals like itaconic acid or 3-hydroxy propionic acid, improving carbon monoxide consumption and production efficiency. This approach demonstrates how engineered strains can optimize natural microbial metabolism. Let us now discuss about computational modeling of microbial consortia. This is revolutionizing the design of synthetic ecosystems, bioprocess engineering and environmental biotechnology. By shifting from species-centered to function-centered modeling, researchers can design robust, efficient microbial communities for applications in bioremediation, bioenergy, agriculture and medicine. So, this organism-free modular approach includes microbial species which are viewed as chassis containing essential metabolic pathways, and they have functional roles like metabolite production, nutrient provision, antimicrobial defense, and these are treated as independent modules that can be computationally optimized. Then databases of these functional modules could resemble biobricks or the registry of standard biological parts for synthetic biology. Computer-aided engineering cycles in silico and in vitro integration. So, here the gears represent the synergy and continuous engagement. Target molecule and system identification is important. We define the functional requirements, example what is the cost and environmental conditions. Then we go for computational matching, search database for microbes with relevant metabolic functions. Then we also undertake mathematical modeling by simulating microbial interactions using constraint based modeling and network expansion techniques. And finally, we validate this model, construct and test the design microbial consortia. So, there are various steps involved in it and also various resources required for carrying out this computational modeling for microbial consortia, but these actually helps us in developing very efficient consortia. So, there is the predictive modeling for synthetic biology. So, here you have the microbiome modeling toolbox 2.0, which is the efficient tractable modeling of microbiome communities. Dynamic models guide synthetic design similar to engineering blueprints. So, some examples include microbiome modeling as suggested by in the systems biology paper. Then MyComs and comets which simulate microbial community dynamics. Then there is the evolutionary game theory and the meta-omics approach which are being explored to predict microbial interactions and optimize synthetic consortia. So, the comets platform in this figure you can see enables virtual experiments by simulating microbial ecosystems in different environments with various biochemical inputs. So, here you have these environments and these inputs, then metabolic models, then we have batch cultures, continuous culture, then medium composition, petri dish, natural environment and extracellular environment. Users can generate scenarios through provided interfaces which feed into the comet's core engine, as you can see over here. This engine models spatio-temporal dynamics, producing data on microbial biomass, metabolic fluxes and media concentrations over time. The results can be further analyzed and visualized using built-in tools or external software. Another important thing is the flux balance analysis in microbial consortia. This FBA is a widely used computational approach that predicts the flow of metabolites through metabolic networks. By applying FBA to microbial consortia, researchers can simulate and analyze the metabolic interactions between different species. This approach helps in understanding how these interactions affect the overall functions and stability of the community. So, there are different classes of FBA: community flux balance analysis CFBA and dynamic flux balance analysis DFBA. So, in community flux balance analysis, and this is an extension of FBA known as community flux balance analysis, developed to study the metabolic behavior of microbial communities. It integrates the metabolic capacities of individual microorganisms and considers the interactions between species and their environment. This method allows for the prediction of community level metabolic fluxes and the exploration of how different species contribute to the community's overall function. In dynamic flux balance analysis, while traditional FBA provides a static snapshot, dynamic flux balance analysis incorporates temporal changes, allowing for the simulation of microbial community dynamics over time. This approach is particularly useful for modeling environments where conditions change, such as in bioreactors or natural ecosystems. So, there are tools like the dynamic community FBA package that facilitate the implementation of DFBA in studying microbial consortia. Let us now move into the third section, where we discuss the applications of microbial consortia in industrial biotechnology. For example, in the case of food and pharmaceutical industry, in the case of power generation in MFCs, and for bioremediation. So, these microbial consortia has various applications. For example, in the production of biofuels and biochemicals, it enhances biomass degradation, then improves hydrogen and methane production. In the case of pharmaceutical and biotechnology industry, it helps in producing antibiotics, support drug delivery. In the case of bioremediation and waste treatment, it degrades hydrocarbons, detoxify heavy metals. In the food and beverage industry, it optimizes fermentation, improves gut health. These are some of the major sectors, but this is not the exhaustive list. There may be many other interesting applications of microbial consortia. So, microbial consortia in food and pharmaceutical industry, particularly for high value products. For example, we have the one-step fermentation of vitamin C. Traditionally, it is a two-step fermentation where gluconobacter oxidants converts this orbital into L-sorbose. A combination of Bacillus species and Gluniseginum vulgare then transforms L-sorbose into 2-keto-L-gluconic acid, which is a key precursor for vitamin C. In this step, K. vulgare carries out the conversion, while Bacillus species helps its growth by providing essential nutrients. But it has this two-step system has a drawback. For example, it takes long incubation time and the need for sterilization twice, which makes optimization difficult. To overcome inefficiencies in the two-step fermentation process, a synthetic G oxidants K. vulgare consortium was developed. So, this reconstructed one-step process using these consortium was 25% faster than the two-step method used earlier, which is the traditional method. The old method required sterilization each step, which added time and cost. The new process removes the need for the second sterilization, making production more efficient. Instead of competition, Z oxidants and K. vulgare now work mutually, improving overall productivity. Then the application of microbial consortia for power generation in microbial fuel cells, where they convert chemical energy into electricity using microbial metabolism. They offer high energy conversion efficiency, functions at low ambient or low temperatures, and then operate in areas without electrical infrastructure. However, single species MFCs like those using Swanella onidensis MR1 suffer from low extracellular electron transfer rates and limited substrate utilization. To address these limitations, engineered microbial consortia are designed to improve microbial fuel cells performances. So, here in this figure we see a synthetic 3-species microbial consortium for high performance microbial fuel cells system. Gymophytes act as fermenters, providing energy rich compounds. Then we have these electrogens which utilize these compounds and facilitate electricity generation in the MFC system. So, here we have Gamified 1 and Gamified 2 and electricism, they are together the 3 species of this microbial consortium. So, you have these E. coli as oneidensis co-culture, E. coli ferments xylose into format and flavins feeding S. oneidensis for enhanced bioelectricity production. However, biofilm formation on the anode reduces efficiency. Then we have the Saccharomyces cerevisiae as oneidensis co-culture replacing E. coli with Saccharomyces cerevisiae prevented anode biofilm formation. Engineered Saccharomyces cerevisiae metabolized glucose into lactate instead of ethanol, optimizing glucose fed MFCs. Then comes the 3 species consortium, E. coli, Bacillus subtilis and S. oneidensis. Here Bacillus subtilis supplied riboflavin as an electron shuttle for S. oneidensis, enhancing electron transfer and overall system efficiency through a division of labor approach, as already shown here in this figure. Then there are also other very important emerging applications of microbial consortia which are trying to address current challenges, for example, like the degradation of plastic, which is now a huge environmental issue. Cow et al. Reported a synthetic microbial consortium designed using a bottom-up approach, with each strain design assigned a specialized metabolic function. Engineered microbes like B. subtilis, Pseudomonas putida and Rhodococcus enhance degradation efficiency by expressing plastic degrading enzymes. This modular strategy minimizes metabolic burden on single strains, improving robustness and efficiency. Converting plastic breakdown products into valuable bioproducts such as biofuels or biopolymers through engineered metabolic pathways is now the recent trend in research. So, you have these plastics which may be PET, polyethylene, PVC, PU, PP. Then you have this strain 1. So, this will attack the plastics and undergo bio deterioration. Then we have these further released into much smaller fragments by strain 2. So, this is an overall scheme of degradation of plastics by synthetic microbial consortia, and then we may have other strains 3, 4 and 5, each of them doing different specific tasks and thereby degrading the polymers into different monomers, and also in the process releasing water and carbon dioxide gas. So, let us now look into the recent advances on microbial consortia for bioremediation. So, let us look into the various substrates like plastic, petroleum hydrocarbons and antibiotics, and what are the achievements in the bioremediation front using microbial consortia. So, plastics like PET has been seen that there is a weight loss of polyethylene terephthalate which reaches around 23.2% in 7 days, and for these co-culture of Rhodococcus, Pseudomonas putida and 2 metabolically engineered Bacillus subtilis species has been used. Then in the case of polyethylene, there was a demonstration of around 81% reduction for LDPE strips over 120 days, and Enterobacter species Bengaluru, and then Enterobacter species Bengaluru BTDSC 0102 and of course 03, and also Pantoa species were used in this degradation. Then there is a polystyrene which demonstrated 12.4% weight reduction of and 23% weight loss of Hips film in 30 days utilizing Bacillus species and Pseudomonas species. Then in the case of polyurethane, around 50% of proprietary aromatic PEPU-A copolymer was consumed in 25 days, when a co-culture of rhodobacterials, rhizobials, brucellarials and then actinomycetils and sphingobacterials were deployed. Then in the case of petroleum hydrocarbons and alkane, synergistic rate of diesel oil biodegradation was achieved 85.5% utilizing Pseudomonas tudgeri and the Gia species. Then in the case of polyacrylic aromatic hydrocarbons, when Spingomonas, Pseudomonas, Spingobium, Dogdonella and Lutemonas were used, it was seen that there was a near complete degradation of fluorine and phenanthrene after 5 days. And experiments on antibiotic degradation, also bioremediation has also been conducted, where sulfonamide antibiotics were found to be degraded up to 78% after 4 weeks by firmicutes and bacteroids when they were co-cultured along with this antibiotic. So, with this we come to the end of today's lecture. Thank you for your kind attention.